happens na? I usually forget to record the class.
Sir, can
Please,
you please repeat that book name, sir, for cell something cell?
actually, there's book name is Pandey. Pandey book for graph physiology. SC Pandey
Okay, sir.
something book will become. Either if you read Life Science Part 1, Pathfinder book, cell chapter. Okay.
Okay, sir.
Actually, please at every class when class will start, please at least anyone please help me to by saying record the class. Please record the class. Okay.
Yes, sir.
I thought. uh, then after that, chromosomal operations. Then chromosomal operations, loss of Mendel. Loss of Mendel, you should cover numerical portion. Many times numerical questions only from. Like we will read what we will read in loss of Mendel. Can anyone define what is the first law of Mendel and second law of Mendel? What's the- Can anyone define what is the first law of Mendel, second law of Mendel? How many laws of Mendels were there?
Two,
How
sir.
many
Law
laws
of
were there?
segregation and
Yeah,
law of
exactly.
independent assortment.
Exactly. Many sometimes many will tell like three laws were there. It's not the correct answer. There are only two laws are there. That is, can you tell what is the those laws again? Can you repeat?
Law of segregation and law of independent assortment.
Exactly. Can you tell what is the law of segregation? Is there any exception for law of segregation?
Disjunction, sir. Disjunction.
Linkage.
No. Actually, law of segregation is universally accepted. There is no exception actually. Yeah, segregation distortion. There is actually he also mentioned the assumptions also. Can you tell what is the assumptions for the first law of Mendel? Okay, leave it exceptions. What are the assumptions for the laws of first law of Mendel? Assumptions.
All the progenies live, sir. All the gametes
Yeah,
produced that will survive. All the gametes produced that will
exactly,
survive.
exactly. Next law, next
Equal
assumption.
frequency of gametes.
Exactly, equal survival and equal frequency. And another thing, equal mating chance, random mating. Okay, if
Yes,
there
sir.
is a assortative mating, you can't get equal segregators. Okay, those things like when we cross P1 and P2, like A, something is happening. Wait, I will discard. I will restart the presentation. When you cross P1 and P2, it should be having 50%. It should be having 50%. Means equal production of gamete, like equal presence of the gametes and equal survival. Here also 50%. Here also 50%. Like sorry, like exactly. Then equal it should mate the A should mate with capital A or small a, and this small a have an equal chance of mating with capital A or small a. This is called as random mating. This is actually one of the important assumptions. Then, as you said, equal survival of the gametes and equal frequency of the gametes. These are the basic assumption for the Mendel's any laws. Okay, not only a first law, any laws. But as we said, like assumptions need to meet, but there is no exception for the first law, law of segregation. In law of segregation, what is the law of segregation? In F1, there will be heterozygous. Can anyone tell what happens in the law of segregation? Define law of segregation. Not explain the concept, define. Law of segregation.
So the two alleles of a gene in F1 undergo
Exactly.
separation and during gamete formation, and each allele uh, enters into separate gametes.
Yeah, exactly. See, what you said is 100% less. It's also called as,
Law of
you
purity
are
of
right.
gametes.
Yeah, exactly. See, why? Because if there is, if why it is called as law of purity of gametes, that's why actually, because we are all MSc students, teaching things, it's like a little bit embarrassing for me. That's why I'm not covering these topics. And one thing, Kaushal, see, when the alleles will there in the heterozygous condition, it will not, this particular capital allele will mask the expression of the recessive allele, but not have any interaction, like it's having interaction, but the purity of the particular allele will be maintained. When they are segregated and passed through as a separate gamete, their effect is not affected by this one. Like that's why they may the when the, even the alleles in the heterozygous condition, the purity of the particular allele is maintained. Okay, that's why it's called as law of purity of gametes. Okay, I think everybody got the point. Anybody have a doubt in this?
No, sir.
Yeah. I have any like my flow is good or like it's like little bit faster, little bit sluggish. Either it
It's
is a
good, sir.
medium.
It's good.
Yeah. Okay.
Sir, one doubt.
Yeah, you can.
Sir, why the disjunction of X chromosome is not an exception of law of segregation? How can we explain that?
Yeah, actually, this see, he's telling when there is a law is a heterozygous, it is separated. The gamete, the allele presence in the heterozygous, it's going as a separate gamete. This is the only the thing he said. He not said anything. Even though you take up extra chromosome, the disjunction it will separate only. Okay, the alleles will separate only, and they will go into different gametes. And XX here will get, and another gamete will not get anything. Segregation is happening, and for example, you are telling Y is there. Y will go one side, and your another gamete will not get anything when there is a segregation distortion. But this particular X allele will not have any influence on the Y allele. Is it correct or not?
Yes, sir. Yes, sir.
Yeah. And after segregation, they will go into separate gametes. Even though it is not receiving any gamete, but it will segregate now. One is the water was like when there is when the alleles in the heterozygous condition, one allele will mask the expression of the other allele, but the purity of the particular alleles is maintained, and they are uh, when they are segregated into separate gametes, and they're having their own effect. Here also happening the same. That's why it is not regarded as exception. Okay, I think now you
Okay.
clear. See, here the allele, this particular allele will not have any effect. Maybe there is a while separation, like while in the meiosis, all the allele go into one pole, the another pole will not get anything. That's why the other gamete will not get anything. But Mendel is not saying that. Uh, that's why he is having assumption, na? Equal frequency of the gametes. In chromosomal distortion, what will happen? Your equal frequency of the gametes will be maintained or not.
Not maintained, sir.
Yeah. Then if we're not fitting into the assumption, it will not need to be say, follow Mendel's segregation. Okay, that's why we are regarding the assumptions. Okay, if this particular law should be there, there are some assumptions. Okay? That's why the law of segregation is a universal. It's nearly universal, you think. Okay, if we feel that that is an exception, it's nearly universal. But law of independent assortment having a severe exception. What is that exception?
Linkage, sir.
Yeah, exactly. Linkage is the one of the severe like one of the major exception for the law of independent assortment. But at that time, Mendel knows, but he not reported anything. He got linkage. Actually, Mendel's another thing. In the next class, when we'll start the actual lecture series, I will give brief. My first topic is Mendel's law. There, I will discuss in detail what are the things we need to read for the SRF exam regarding Mendel's law. Okay. Then, cytogenetics. I will cover in detail about polyploids, how to calculate the numericals in polyploids. Okay. Then gene interactions. I think what is gene interaction?
So interaction means interaction between two alleles of different genes.
Interallelic interactions.
Yeah, exactly. Two alleles of a different gene. Not only two alleles, any alleles of a different gene. Exactly. There are types of interaction. Many of you know that complementary gene action, supplementary gene action. Okay. Those things actually I will not go into detail. Actually, Mendel's law will go in detail for the because Mendel's law need to be like what is the molecular characterization of Mendel's. There is a recent paper was published. I will discuss the paper in the next class. And cytogenetics I will go in detail. But gene interactions and history of genetics, this chapter I will not taught in this course. Okay? As I mentioned in the course list. And quantitative genetics, I will deal in detail. What is quantity? For example, if somebody is working on resistance, somebody told, okay,
Yes, sir.
somebody is working
Myself.
on what is your disease like?
Sir, insect resistance.
Yeah. For example, your resistance is there. You want to know that resistance is quantitative or a qualitative. What experiments you are going to conduct? You want to know your resistance will be there, like insect resistance. You want to know either the resistance is monogenic or oligogenic or quantitative. How will going to know what experiment you will going to conduct? Can anyone? Not only him. Can anyone also respond? Chart 9.
Sir, if F2 gives us normal 3:1 ratio, then we can decide it's monogenic. It is controlled by monogenic. If there is three susceptible reactions or one resistant reaction or one susceptible reaction, three resistant reactions like that, we can detect monogenic. For oligogene, if the dihybrid cross ratio is came, then we can confirm it as oligogenic.
Yeah, exactly.
But
Then
for the quantitative, there will be grading in the resistance.
Yeah, quantitative. Can anyone exactly define like what we will get? Yeah, as he said.
Symmetrical distribution of characters.
Yeah, exactly. Normal distribution or symmetrical distribution. Okay, you will get a continuous variation. Like it will follow a normal distribution if the resistance is governed by a quantitative trait, like quantitative many minor genes, then you will follow a normal distribution. If monogenic, we will get a clear classes like 3:1 ratio, as he said. In oligogenic also, we'll get a dihybrid cross ratio. That's we had to analyze or we had to screen the population, either F2, segregating population F2. Then we will come to know that okay, the particular trait is monogenic, oligogenic, or quantitative trait. So I will go into in detail what are the experiments were conducted and what quantitative traits are follow Mendelian laws of segregation, Mendel laws of principle of heredity. Either
Yes,
it's
sir.
not follow the yeah.
It follows.
Why
Sir,
it is following?
the basic underlying concept of segregation are everything same, sir. But here interaction plays the role, sir. Polygenic
Yeah,
interaction.
exactly. Yeah. Who proved that? Who proved that?
Nelson and Cayley, sir, I think.
No, no, no. Nielsen LA. Yeah, Nielsen explained the actually, but exactly the precisely he said that the Mendelian's like quantitative trait will follow the Mendelian inheritance. Actually, the yeast experiment of corolla length of tobacco. Like I think tobacco corolla length, he crossing the medium long corolla length, short corolla length, then he will get an average corolla length in F1, then F2 segregation, F3 segregation he analyzes, then he come to conclusion that okay, the quantitative traits are inherited with the minor genes, but they follow Mendelian principles of heredity. Okay, that
Yeast
we will
experiment.
discuss in detail. Yeah, yeast experiment on corolla length of tobacco. Then another experiment we will discuss Nielsen L experiment, yeast and corolla experiment, corolla length experiment in detail, and also cover quantitative and biometrical genetics in detail, no doubt in that. But I will not cover basic things which are you are reading from the BSc. I will not going to cover. If you have any doubt, please contact at any time. I will go. I am free to discuss. Then linkage. This is a very important. Why? Why it is so? Can anyone tell what can be done with the use of linkage and recombination information?
QTL mapping.
Map, genetic maps.
Yeah, genetic maps. Okay, genetic maps. Then QTL mapping, the GWAS mapping, maps. As he said, mapping of a gene. If you want to map any gene through any method, the basic principle, underlying principle is linkage and recombination. If you not have any clear understanding about this linkage and recombination concept, you will not going to understand the entire molecular breeding, entire marker systems. You will not going to understand. That is the basic chapter I will going to deal. How we can use a linkage and recombination information. What exact through for mapping. What is the first genetic map is created? Like who created and what is the first genetic map? In which organism is created?
Drosophila.
Who created?
Stuart and sir. Stuart and
Yeah,
student of Morgan.
exactly. Yeah, student of Morgan, as he said. This is exactly right answer. What is mapping functions? Why we need to use mapping functions? Can I extend another 20, 30, 15 to 20 minutes? It's okay for you all?
Okay, sir. Okay.
Yes, sir. Okay.
Okay. Then what is Halden mapping function and what is Kosambi mapping function? Why first question is why we need to use mapping function? I will discuss in detail like day after tomorrow.
Sir, after certain percent of recombination, the distance is not clearly estimated on the chromosome, sir. So we have to use mapping
Exactly.
functions for.
Exactly. Yeah, as I said, what will happen when there is a more distance? What will happen?
When there is more distance, then recombination will happen, sir. Or there will
What
be
exactly the term? The term you're missing. I was
Crossing
whenever
over.
the
missing or.
yeah no another term you are missing. Multiple crossovers. Okay, double crossovers, multiple crossovers. What is the formula for coefficient of coincidence?
Sir, 1 minus interference, something is there.
No. Like, okay. Formula 1 minus interference is also okay. Exactly. How will calculate interference? Then what is interference? See, when the interview, like any interview you go, if you give any key term, that's a special term, then they will ask the question, the cross question. What is interference? Actually, we are making the interview difficult. Actually, when we go for the interview, actually, as my
Interference,
senior
we
said,
can define it as the happening of a crossing over prevents the happening of another crossing over. We can say it as interference.
exactly. That is. But to avoid interference, okay, interference is one factor. And another factor is as the multiple crossover will happen now, your genetic distance will not correlate with your recombination frequencies. That's why we need to use mapping functions. First, Halden used the mapping function, but he not even taken the interference effect. Means it's overestimated the effect. That's why, but in nature, interference will happen. That's why Kosambi included the interference and he refined the genetic distance. Okay, that's why when there is a multiple crossovers, as you said, there is a more distance. The recombination frequencies will not correlate with your mapping distance. That's why we need a mapping function to correct it. There are two mapping functions, Halden mapping functions and Kosambi mapping function. In Halden mapping function, we will not include interference, but Kosambi mapping function, we include interference. If you know this much is more than enough, but what exactly this means, I will going to discuss in the future classes. Then the mutation. Mutation is also a very important chapter. What is exactly the recent trending? Actually, I made a one video. What actually recent trending in mutations? Which mutant is revolutionized the green revolution? Can anyone tell? India is the one of the top researcher, especially in the mutation breeding. Some are, I think, many. One of the, I think, Dublin, I think, is screening mutant population for some traits. Okay. Why we are going for the mutations? Searching for spontaneous mutations or searching for like creating the induced mutations. Why we need to go for the mutations in search of mutations or in creation of mutations? What is the need? Can anyone tell?
It's the ultimate source for variation.
Exactly. We're actually hitting the points. Okay, it's very good actually. Then ultimate source of variation. Okay, that's why if there is no variation present, ultimately either we search the natural mutants or we create mutation. That's why it's a very important chapter. Whatever the diversity, so many are doing variability studies, diversity studies. The variability exists because of there is a mutations in the particular gene. The gene having an alternate form of the allele. You are counting that what are the number of alleles present in particular species. Itself is the diversity analysis. What you will do in diversity analysis? You screen for a molecular marker or you screen for a phenotype. If there is a different phenotype means there is definitely a different alleles. Then again one question. If for example, if there is a two traits, for example resistance only, if you want to know either for example one species A, species B, both are resistant for rust. If you want to know either the genes governing resistance for rust is same or not, they are allelic or not, how are we going to check? Either they are alleles of a same gene or they are separate genes. How are you going to know? What test you are going to conduct? Actually, this class is mainly to check
We,
your knowledge actually, so that I
we
can
need
plan my.
virulent pathotype for that, sir.
Can you repeat?
We need a virulent pathotype for knowing that.
Yeah, that also done. But basic genetic experiments you can plan. What I want to check is this
Sir,
alleles or not?
there will be recombination between the two genes if they are from different locus, sir.
What exactly the tests I'm asking? You are on the way, but I'm asking the exact the word. Which tests?
Complementation test.
Exactly. Complementation tests or allelic test, allelism test. Okay. If you cross them, then you are getting wild or a mutant. Based on that, we're going to decide either they are allelic in nature or they are different gene itself. Okay. Based on that, we're going to discuss what exactly the complementation test, what is recombination test, how to uh, how to take how to map how the classical definition of a gene was designed by these experiments. We will discuss in detail. Okay. Then okay, these are mutations actually. I will not go in detail about the mutation breeding, but I am dealing with the side-directed mutagenesis like SD1, SDN1, SDN2, SDN3, and how it is useful. Okay. Then population genetics, it's a very important. This year, actually, what happens now? Last year, up to 2022, 23, nearly three papers majorly focused on molecular breeding and basic breeding. Majorly focused on molecular breeding, and then my teaching is because I was my MSc was completely molecular breeding. Now also I'm in molecular breeding. In ICRISAT also I was doing molecular breeding only. I was good in I was somewhat experienced in molecular breeding, but I have not teached many things from the population genetics or biometrical genetics. Actually, you can find any different platform which are like either any. There are other two platforms are teaching SRF. If you join there also, you see me only because I only recorded the lectures in the both the platforms. And now they are selling for I think some amount. Okay, now everywhere you join, you see me only. But I focused there only on molecular breeding, especially. But the question paper in 2024 is majorly on population genetics. And normally we neglect this course. Actually, we will not take course as population genetics course. But in IRA, they will teach like anything this course. But the course teachers are very tough. That's why we neglect. Will not take this course. But this year, the all the questions from this course only. Like that. There's some issue has happened. That's why maybe they have. Population genetics is a very important thing. I will actually from Dharwad, those who are from Dharwad, for them it's a very easy because SS Patil sir is taking like excellent classes. But for us, even from IRA or any other university, these population genetics and biometrical genetics is very tough. But I will try to deal because I will also prepare well before coming to class. Actually, they are all formula based population genetics. I will try to teach my level best. Okay, I will be in detail in population genetics. And these things, DNA replication, transcription, translation, repair regulations, I will not go in detail and I will not teach also. Okay. Then marker, because these things are either I teach, either you read, it's the same. You need to buy it. Okay. Either so much videos are available for life sciences, like there are so much like Somish biology was there, Jyoti Kumari videos are there. So much things are available. You prepare from there. Okay. It's not necessary to teach this. I will focus on other things which are very important. Markers and marker assisted plan. This I will go in very detail. The QTL mapping, Jivas, reverse breeding, whatever it may be, in the genomics plant breeding, I will going to deal in detail. Don't worry about that. But what are the basics I am highlighting which I am not going to deal? Please read. It's not like that. It's not important for exam. Why I'm avoiding this means even I teach or if you read, it's the same. Okay, there is no like concepts in that. You should mug up and you should remember and you should forget after this. Then biotechnology. Biotechnology also, you have to read one book called as only one chapter from the Pathfinder book. I think nearly 70 to 60 pages are there from Pathfinder book, Parts Life Science Part 2. You read that biotechnology chapter is more than enough for the exam. But you should more focus on markers and molecular breeding. Then I think I hope it's already 1 hour or like 1 hour 20 minutes. This is the thing like I want to introduce myself and I have to take the introduction from you and just I uh, highlighted which course I'm not going to deal and which are the topics I'm not going to deal, which are the topics I'm going to take. Okay. If any have any doubts about this class, I will wind up the class now. If anybody have doubts and suggestions, please suggest today only. It's not only today, but today if you suggest means I will plan accordingly. I will ask one by one. Saukarya Das, sorry, Saukarya Das, you not introduced, na? You left. Okay. Anybody have any doubts? Sumit, you are in first year. You should listen. Okay. Yeah. Sunandan, how are you?
Hello, sir.
Yeah, how are you? Yeah.
Sir, I want to ask one question. My friends, some friends are going to join our course. If
Yeah,
they
yeah,
can
yeah.
join, I want to ask if they can join today or tomorrow.
Yeah,
Tomorrow, day for
hey, no, no, no. Anytime they can join. Only problem is they have to watch recordings. I can't repeat the live class again and again. That's the problem. They can join at any time. At the even after the end of the course also they can join. But they have to listen the recordings. Okay. It's not like that today. Yesterday was the last date. It's not like that. But I suggested if you are listening to live class, it will be very helpful because you can interact and I can also answer better in live classes. Okay, you can suggest. Any doubts? Any doubts?
Sir,
Anandan or
as you told about your previous classes from any other platform,
yeah, yeah,
whether
yeah.
they are same to our classes that we are going on now, or whether they are different from, because we
No,
can
exactly
also
same.
get there and watch those videos too, sir, if you say.
No, 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 no. Same because I am the person the teaching everywhere. Actually, maybe see what everyone is doing now. What are the things which are specifically for SRF? I already made the videos actually because see the cell division. What you will go see everything is available in the internet. But the things, if you are searching for these things which are specifically for the SRM, like molecular breeding you take, genomics in plant breeding you take, QTL mapping you take, these things are not available anywhere in the internet. Is there, but they are so complex. But at student level, at particularly for SRF, nowhere it is there. But there also, I only teach. I think Sunandan was in Krashi and Pavitra is in Agredict because uh, in both platform I teach the same. If you wish, you can join, but the same lectures will be there. You can hear also. But it's not like that. I kept 1,500. It's I will deal very short. Like it's not like that. Actually, I have normally made classes based on the per day. Normally, I will teach based on the per day lecture basis. I don't know how many students they will going to join and what the price they will keep. I was not worrying about that actually because I will be allotting for the particular topics. I will dealing with that and I will wind up in the course actually. But the thing is, after but I am feeling that to listen my classes, you need to pay that much money. I don't feel like it's worthy. That's why I started with this one. Actually, I started with the free, but I had to subscribe the channel. I had to buy this iPad. This what is this writing pad and internet access, accessing the internet, mic, everything. It's like subscription. It's a very big thing actually. But it's not like that. I'm giving very less content. It's also a revision for me for ARS. Okay, more I will teach, more I will become precise, more I will write in the exam. Like I will write in a very nice manner in the exam. It's also helpful for me also. Yeah, I will teach in detail. If I teach in half of things, na, I will also learn half of things, and I will write like my ARS will also go on. Going to fail. That's why I will be in detail. Okay. What are the concept? The 70 lectures I told now, it is more than enough for the SRF examination. Okay, no need to take any coaching. Even I suggest don't take this coaching also because if you have got proper guidance, if you have like for example you have a one senior is very good in subject or very good in guiding you, no need of coaching. You just ask him what to read. If you have doubt, you ask the question for him. He will solve. This is how actually it will go. Okay, those students who are like who are joined in any other platform, those who are interacting me continuously, like sometimes they should text me at any time. Okay, I think there many are there. I think they are getting what I'm saying. Those students are already in higher, and those are showing good rank in their respective states, and even they got better rank actually. It's not like that. If you have to take coaching, then only you will clear the exam. It's not like that. You need guidance because you already crossed the stage of somebody has to there to teach you everything. You already crossed the stage. Okay. You just need to know what is the things are there and how to read it. Okay. If you're not understood, putting chat GPT, it will tell the near very good answer. Okay. No need to pay that much money for this SRF. Okay, no need to go any coaching. Even I suggest if you have very good senior and if you have a grip on your subject, no need to take coaching. Okay, any coaching, not my coaching, any coaching. For plant science, what happens now? In UG, like many colleges don't have proper teaching. Okay, they need at least because they are so many they enjoyed they like will they normally up to third year they will enjoy. Fourth year will start to read and like this will happen. That's why we need some coaching notes, script notes, those things. But SRF is endless. The syllabus is endless. Whatever they can prepare. Okay. You have to read your own. Okay. And you need guidance. How to read and how much to read. Okay. No need of any coaching. For after MSc. For any exam. For CSR, for SRF, for ASR Binet. No need of any coaching. Okay. Any doubts? I think what I'm telling, what I'm want to convey, you got, I think. Hello?
Yes, sir.
Yeah. But one thing I can clearly tell, wherever you go, you can find me only. And maybe if they're teaching extra, but all those are like they're teaching for plant science students, they will allow you to join there only. And you have to listen with, along with them from basic details. But my suggestion, go in n number of channels for this. Like if you want to read for the genetics, you just type genetics CSR. So many videos will come. Excellent teachers like Somush Biology and Jyoti Kumari and Bansal Biology. There are so many things, sir. I actually watched their videos for genetics, actually, especially for the genetics. And plant breeding, um, basic plant breeding, I will deal. Okay, but if you are more, you want more, you just click on the, you just search NAHP cast IRA genomics in plant actually genomics in crop improvement, one playlist will be available. If you go through that playlist, all the teachers of IRA have taken the classes. Association mapping, my guide is expert in association mapping, Ranjit Kumar Ellur. You can watch his videos, you'll get very each and every point of association mapping. Util mapping, Akshay Talukdar has taken, KK is my member, has taken, he is also excellent in biometrics. He only teaching us in IRA. He only take class in that video is available in YouTube. And for reverse breeding, and for example, transcriptomics, he only teaches, he has already made video on YouTube. There are playlists available. If you want, you can watch them also. Okay. Don't take simply a coaching, these things are bogus things. Okay. Anything? That's why actually I started this course. Actually, I was not interested to start actually. Yeah, then to avoid joining with paying a lot of money, I started this. Okay, anyone, any have doubts, you can tell, can ask. No doubts. Can I wind up?
Yes, sir.
Yeah. Okay. And today I think other than class, I talked other things lot. Sorry. From tomorrow I will be very stick on to my topics. Okay. I already have presentations. I will deal with them. If you have any doubts, please query. Like you can ask me at any time. Okay. Okay. Bye. Bye everyone. Thank you. Thank you for attending my classes. Thank you everyone. Bye.